We are so excited to be hosting Dr. Brian Beal tonight. He teaches courses in marine biology and experimental design and conducts research on commercially important shellfish at the University of Maine at Machias. And tonight he's going to be talking to us, as you know, about softshell clams in a changing climate, how seawater temperatures and predators regulate populations along the Maine coast. So Brian, I'm going to pass it over to you. Thank you so much for being here.
Hello?
Oh, hi, can you hear us?
I can. You keep going in and out, so I don't know what was happening. Am I ready to go?
Oh, I apologize for that. Yes, you're okay. Ready to go.
All right. Well, here goes nothing then. So is my screen being shared right now? Okay. So hi, everybody, and thank you very much for joining us. My name is Brian Beal, and I work and teach at the University of Maine at Machias. In a few weeks, actually, this will be my 36th year of teaching and doing research here at UMM. So tonight's talk is about softshell clams and what's happening to them in our changing marine climate, specifically how temperatures and predators are affecting or regulating populations along the coast. I thought I'd first start with a little bit about the life cycle of the softshell clam. And clams have separate sexes, and they produce eggs if they're females and sperm if they're males. They have external fertilization, and so the eggs and sperm are placed into the water column or maneuvered into the water column. And let's see, I'm supposed to be able to, a little bit of technical difficulty here. Let's see. Well, it's not quite working the way I thought it would. Let me see if I can, no, it's not gonna work, so yes.
I don't know if this would make any difference, and maybe Jake can help, but you're not in presentation mode, I don't think.
Oh, okay. I thought it was, sorry.
No, that's okay. We can see your screen, but we can see like all the slides on the left-hand side as well.
Perfect, okay. Well, there we go. All right, well, thank you very much for that. So when softshell clams spawn, they place their eggs and sperm into the water column where fertilization occurs. And a few hours later, depending upon temperature, a swimming organism called a trochophore is looking for food and being maneuvered around by currents. Now, this animal is about 40 microns, which is 41,000th of a millimeter. So it's quite small. And through the next few days and into some weeks, depending again on temperature, it starts to develop and it forms a shell. The shell is removed in this picture of the veliger just to see what the internal anatomy looks like. But the main organ is the velum, which is its swimming organ that it also uses to capture phytoplankton or small microalgae in the cilia or the hairs of that velum. Just before it is ready or competent to settle, it develops a foot, and that foot is used when it sinks to the bottom to probe the bottom. And if it's a hard bottom, what it will do is it will move its foot back into its shell and then stick out its velum and then go swimming away until it drops out of the water column again and again uses that foot to determine whether or not the sediments they're able to, that the organism is able to burrow into. So after a while, the choice becomes essentially zero because the animal is going to metamorphose into a juvenile. And it is at that size that the juveniles will end up in the sediments themselves. I've got a couple of pictures here. This is a photomicrograph of an 8-day-old clam. This is the veliger stage. It's actually a D veliger. You can kind of see what a D looks like as it rotates right there. The major part, of course, is the velum, the swimming organ, but you can also see that dark gut in the middle, which means that it's been feeding on phytoplankton and is just simply growing and swimming. And I'll show you where this is occurring in just a minute. But to give you a sense of how big that animal is, this is a scale. The upper part is 1 millimeter, and the bottom part is in microns. So there's 1,000 microns in 1 millimeter. And the size of the animal that you just saw, the 8-day-old veliger larvae, is about 100 microns, or 1/10 of 1 millimeter. After about 3 weeks, these animals are settling to the bottom, where actually they're smaller than most grains of sand. They're about 200 microns, but they're fully equipped. They've got this foot. You can see on this glass slide, the animals just crawling around. And again, if we use the same measurement, this animal is about 200 microns, so it's doubled in size over the course of that those extra 2 weeks. A little bit more about clam biology. Again, clams have separate sexes, and the gonad isn't a separate organ like a scallop, but it's in the tissue in and around the gut. So during times of year when the animals are ready to spawn, that gut becomes this very beautiful creamy color. And when you poke it, it's very plump or turgid. And so this is what a male clam would look like releasing its sperm. And this is a female. The eggs are not buoyant at all. They kind of sink to the bottom of any vessel that they're in. And so the eggs need to be up in the water column. So because of currents and storm events and things like that, these eggs get into the water and then are fertilized. It's a messy thing and it's not very efficient. 1 2-inch softshell clam female can produce about 2 million to 3 million eggs, and 1 2-inch male can produce around 10 million sperm. So if one of those eggs makes it to reproduction, then we have a stable population. So, you know, this is a very sloppy way of an organism evolving in such a way so that it carries on, but it's been successful. It's millions of years old. If we look at the relationship between the weight of the gonad versus the weight of all the tissue, we end up with something called a Gonad Index. And we can see from this graph that there's special times, certain times of the year, in the late April to late May timeframe, when the Gonad Index is highest, which means that it's ready to spawn. And then when seawater temperatures reach about 10 degrees Celsius or 50 degrees Fahrenheit, the clams will spawn and they will lose that gonad. And this is what they will look like prior to spawning. And this is what the animal looks like. It's not quite as plump and turgid in terms of its gonad as it was when before it spawned. So as seawater temperatures begin to cool off in October, November, and December, it's putting its energy into reproduction rather than the production of shell. And we know about this because we've been spawning clams at the Down East Institute, and its former not partner, but its former iteration called the Beal's Island Regional Shellfish Hatchery since 1987. These are pictures in the microalgal grow-out area at DEI today, and this is our mass culture area. And so we use this algae to produce larvae or to feed larvae, as well as to condition softshell clams to spawn. And this is all done at our new facility at DEI, which is in the town of Beals on Great Wass Island. And hopefully we'll be having a shellfish field day this summer in August, I'm hoping anyway. But to just give a really quick version of how we produce clams, and I'll be talking about some of the experiments that we've done using these small clams. We obtain animals from the wild. We stimulate them to spawn by taking them from 50-degree Fahrenheit seawater, and warming it up to about 72 degrees, which is about what's happening on flats in the springtime. If you went out on a flat in May at noontime and it was low tide, that brown mud is going to heat up. And when that water comes in just a few millimeters initially coming back over that warmed up flat, and you measure the temperature, it might be 40 degrees in the air, but if it's sunny, you might measure seawater temperatures as warm as 80 degrees only for a few seconds. But that change in temperature is one of the factors that affects clams and initiates spawning. Again, a 10 to 12 day old veliger. We do these in large 400-gallon tanks at DEI, feeding them different species of phytoplankton or algae. And our goal is to get them to a size where they'll rest on window screening, which is about 3 millimeters. So we're looking at an animal that's 0.2 millimeters when it settles, and we're having it leave the hatchery when it's 3,000 microns. So 0.2 millimeters is 200 microns, and 3 millimeters is 3,000 microns. So these will sit on window screening, and we place 10,000 of these animals at a time into trays that float on the surface of a cove on the opposite side from where DEI is, in a cove called Mudhole Cove. And over the course of June to November, these clams grow from about 3 millimeters to a little bit more than ½ an inch in size. And so this is what a tray that's 3 foot by 4 foot would look like. Whoops. We throw a handful of periwinkles in each side of this tray, and they're like little cows. They're grazing, and they keep the aperture of the window screening from getting fouled by macroalgae like kelps and things like that. So we end up with, lots and lots of these very small seed clams, and that allows us to do a variety of things. One of the things that it has allowed us to do is some fairly applied research on how they grow and when they should be planted and things like that. And all the experiments that we've done, which are fairly small scale, you can see here that my experimental unit is a 6-inch plant pot. But a lot of the results that we've found at this scale have been applied to commercial scales and scales that communities are using. One of the things that we first noticed that when we planted these clams in the field in mudflats was that they marked themselves at the same time that they're planted. So regardless of whether or not they're a 1/4 of an inch or an 1/8 of an inch or ½ an inch, they produce this disturbance line at the time in which they're planted. So it allows us to do a couple of things. One, to distinguish a hatchery clam from a wild clam. And second, it allows us to get a growth rate off an animal that we didn't have to uniquely mark. So every individual that survives provides us with information about how well it grew. And so when we put these clams in different habitats, whether or not, I'll go back one, they're in eelgrass, as this shot shows, or adjacent to eelgrass, or whether we put them at the high water mark or the low water mark, we're able to get a really nice estimate of their growth that's recorded in their shell. In other words, here's a shot that shows some clams that I planted at that flat in Cutler in May of 1989. And you can see that I actually planted them in April, and they were much smaller than this. And you can see the little round white rim of those clams in May. So let's go ahead a month, and we can see how fast these clams are growing between May and June. Now let's find out how fast they grow between June and August. And it's incredible because it turns out that about 60% of the yearly growth of clams is occurring in this very short, narrow window of time. If we were to look at this using this bar graph, we'd see that about 5% of the growth occurs between April and May, 15% occurs between May and June, 60% between June and August, and the rest, about 20% between August and October, which leaves about 6 months of the year where the clams aren't growing in shell, but they're actually growing in terms of their reproductive capacity. Excuse me, they're putting energy into reproduction. So if we go back to some of these very small scale studies, these plant pots allow us to do a variety of things. put different kinds of sizes of nets on the pots, leave some open to determine the effects of predators. The lower left-hand corner shows clams that we planted in the upper intertidal of Mudhole Cove. We put them out in November of 1988 and went back in July of 1989. And because of the other studies that we've done, we know that they didn't grow anything between November and April. So all that new growth that you see, that is from the hatchery mark out, shows what they grew from April until July of 1989. We've been learning and continue to learn about the effects of predators. And I'll show you talk a little bit more about predation in just a bit. But the upper right-hand clams, these were clams that I worked with a 4-H group in Jonesboro way back in 1984. It's my first job out of graduate school, and we didn't know about the hatchery mark then. So we were actually painting color dots on clams to be able to distinguish them in the field in future. And we found that obviously that was kind of a waste, but we didn't know that at the time. Those clams in the upper right have been killed by green crabs. The one in the lower left-hand corner is a clam that has been drilled by a moon snail. Euspyroheros is its name, the northern moon snail. And all of these activities have allowed us to go out and work with communities and clammers. They've also allowed us to work with elementary school children and high school kids that we've been looking at. This is one of my favorite shots. It shows absolute chaos on a mudflat with kids from the Cutler Elementary School, and this is what I dream about is having kids learn about the kind of things that makes me excited. And we've got kids that they can be in 3rd grade or 4th grade. They know how to use a caliper and can record on our data sheets and contribute to the overall data. It's been really cool working with them. One of the things that we've learned from these small scale studies is the importance of predation along a tidal gradient. And this is a somewhat confusing picture, but let's pick it out one shot at a time here. Let's go to the high tide. And what we've done is we've put clams out in plant pots, and some of them have been open or unprotected, and others have been protected with netting. And the netting that we typically use is between a 1/6 inch aperture and a 1/4 inch aperture. And what we find at the high intertidal is that there's really no difference, statistically anyway, between in survival between treatments where the clams are protected and they're not protected. And what we also found was that extends to the mid as well. So the heights of the stipple, or the meshed bars, and the heights of the black bars are just about the same for the high and the mid. But when we go to the low water mark, we find that predation is much more intense. And that tells us a little bit about where the predators are coming from, which is from the water, mostly crabs and fish. So these predators are waterborne, and if we protect them at the low, we get just about the same kind of survival rates as we do at the high and the mid. But if we don't, we see that predation is pretty important. And so when you look out on a mudflat, or you take a walk from the upper intertidal to the lower intertidal, you're bound to see lots of holes up near the shore where predators don't get a chance to forage at high tide for very long. And you see lots and lots of holes, and that's where lots of small clams are occurring. And as you walk further and further and further out on those flats, you find typically fewer and fewer softshell clams. And the reason has to do with the number of predators and how long they actually have to prey on clams that are inundated for such long periods of time near or at the low water mark. In terms of growth rates for that same study that we did in Jonesport way back in 1996, the upper shot here shows the effect of tidal height on growth rates, and the black circles show the high water mark, and you can see how much slower those clams grow at the high water mark than they do at the mid versus the low water mark. So there's a trade-off. Clams don't grow very fast at the high water mark, but they survive really well. And clams grow very, very fast at the low intertidal, but they don't survive there very well. In terms of netting, it has really no impact on how clams grow. And we looked at density at the same time, and 330, 660, and 1320 per meter square equate to about 30, 60, or 120 clams per square foot. And you can see that at those densities, again, over the course of the growing season, there wasn't much of a difference in terms of stocking density. So we've used that information to inform how we work with communities to try to enhance clams on flats. And so we use these large 12-foot by 24-foot nets to protect clams that we seed at, again, somewhere between 30 and 60 per square foot. And we do this in Edmonds. We do this in Washington County. We do this actually in most counties. Here's one in southern Maine. And this is a project that we did in the town of Freeport back in 2014, where we planted clams either at 15 per square foot or 30 per square foot in an area that hadn't seen a commercial clam harvested in about 30 years. In fact, the clammers called it dead mud. So, you know, this is where a lot of towns want to enhance their flats because there's no clams there. And in many cases, there haven't been clams for quite some time. So these clams were planted typically in April or May. And this is a shot of early November of 2014. And when we looked at the mudflat surface, we got pretty excited. This is a 2-pound coffee can that we use as a corer. And what we do is we push this can into the mud, and then we stick a rubber stopper in that hole, and we extract the core of mud. And what's nice is that no matter where we are, it's the same surface area. And so we can calculate what the densities are from one place to another. So when we looked at this one, we can see we found actually hatchery clams, again, because they've marked themselves, and that's really nice. The other thing that you notice is how fast they've grown. They started out at these small, almost half-inch sizes, and about 5 to 10% of them actually reached 2 inches in one growing season. Which was fairly incredible growth. But we also found that most of those holes had nothing to do with the hatchery seed. They had to do with wild clams that had settled there, and the netting protected them from predators. We know that because when we went outside of those nets and took core samples, we found very, very few clams. So this was one of the most remarkable results that I had seen. I mean, you wait till you see the next few slides. So we're taking this one net. It's 12 foot by 20 feet. And we know that the average number of recruits from all those cores that we took was about 1,300 clams per square foot. And if you multiply that by the square footage, you end up with an extra number of clams that's almost 1/3 of a million. Well, we didn't have just one net there. We had 40 nets, and we didn't find any difference underneath all of the nets. And so this was a very, again, exciting time because we estimate that there were an extra 13 million wild clams in an area that had not seen a commercial clam harvested in about 25 to 30 years. So this is going off on another quick aside about using small-scale studies to look at what large-scale processes are. I'm going back to that flat in Cutler, and what we have is eelgrass. And some of you might say, well, I've never seen a flat that has eelgrass on it. This is rare. It's rare in Washington County, and I know that McCoy Bay in Brunswick sometimes gets eelgrass, but the mechanism on why there's eelgrass in Cutler versus most other places is different. There's a subtidal eelgrass bed, and the eelgrass is a flowering plant, actually. It's not really a macroalgae or a microalgae. It's actually more related to the plants that you grow in your garden because it produces seeds. It's an angiosperm, and those seeds need seawater to grow and germinate in is the word I'm trying to get here. And so they make these seeds, and then the seeds just simply fall to the benthos and then get washed around. And some of the seeds make it to the intertidal. Well, in most intertidal areas, there's not these tiny, well, tiny, they could be the size of a house or the size of a school bus, or even larger, and these intertidal eelgrass beds. So how do they form? Well, it turns out that the intertidal eelgrass here in Cutler forms because of ice. When ice builds up on the shore and moves across the flat, sometimes it scours, and it scours sometimes these little pools, and then these seeds of the eelgrass fall into these pools and germinate, and then we get these beds. And so what I've done with my classes here at UMM, and dating all the way back to the late 90s and early 2000s, when I was working with a colleague, Bob Vadas, at Orono, he'd bring his marine ecology class down, and we'd set up these, interesting little experiments where, in this case, this schematic is showing that we varied the density between 12 and 24 of these small clams in these plant pots, and half of the pots were protected with netting and half of them weren't, so that we were looking at predation. And then we planted half of the clams outside eelgrass beds in the unvegetated areas, and the other half inside the beds. So this is, again, that same shot that I showed you before. And here's a picture of one of the pots that's, right flush with a sediment surface in an eelgrass bed. So, I'm not really going to talk about the results of that, but I will say that eelgrass and softshell clams don't really go together. The eelgrass is a great place for small crabs to hang out, and we actually found lower survival rates in eelgrass than we did outside the beds. But the reason why I've brought this up is because from 1999 to about 2006 to 7, we were getting, on average, when we averaged all the pots, regardless of how many clams were in them initially, regardless of whether they were protected or not, we got about 58% to survive from April, May timeframe to late October, early November. But something happened in 2006 and 2007, and we noticed that from our data showing a huge decline in average survival. Well, the other thing that we noticed is that in 2006, 5, 4, 3, 2, we weren't getting many green crabs in our plant pots, but through time, especially starting in 2007 and then between 2007 and 2008, we found quite a few green crabs in our pots collectively. And so what was really happening? And when we superimpose the number of green crabs in blue and the survival of clams in pink, there's kind of a correlation between the number of crabs that we found and that declining survival rate of clams. Well, we know from work that Andy Pershing has done and published in the journal Science that for 35 years, starting in the early 1980s, there's been this gradual increase in sea surface temperatures in the Gulf of Maine. And, that's coincided with clam landings. If we look at clam landings from 1950 to 2019, we find one thing, that is that, this is a viable fishery. It's averaged about $15 million annually since 2000, but you can see that there's been two major dips in the landings. One was between 1951 and 1960, and the other one has been essentially since about 1976. There hasn't been much of a change. In fact, 2017 was the lowest annual clam landings in the state since the early 1900s. And since the 1970s, that's meant a loss of about 60% in terms of the amount of clams that have been harvested. That has gone on at the same time where measurements of seawater temperatures out of Boothbay Harbor, just looking at the wintertime temperatures, have shown a steady increase since the 1970s in seawater temperatures, although there's that 2006 timeframe when we think that something happened in Cutler with green crabs. And this is the same graph, just showing a decadal increase gradual increase in seawater temperature over time, again with the 2006 there. So we know that seawater temperatures are increasing. And if I go back to this graph here in the 1950s, it turns out that from 1951 to 1955, seawater temperatures in the Gulf of Maine were almost identical to what they are today. That is to say, quite warm. And that coincided with an explosion of green crabs in that same time frame, and put many clammers out of business, hence the decline of clams. In the 60s and early 70s, we got a lot of ice, and it was very cold during the winter, and green crabs, their populations just simply plummeted. But now they're back on the rise again with the rising seawater temperature. So we're kind of back to where we were in the 1950s with where we are with temperatures and green crabs. And this graph, although there's not a really good correlation, it's not that strong, but what it shows is that you might be able to predict how many clams are being harvested based on seawater temperatures. And so what I've done here is I've looked at the harvest and then gone out 5 years or back 5 years to look at water temperatures. So there's a lag effect that I've looked at here, because it takes roughly 3 to 5 years for clams to grow to commercial size if we average clams along the coast of Maine. So this is just another example of the effects of seawater temperatures, not the effects, but the correlation with seawater temperatures and landings. The last thing that I'm going to be talking about over the course of the next few minutes are some small scale studies that I did down in the Fore River in South Portland with some folks from the Wainfleet School in Portland, and in the Webhanet River in Wells with folks from the Wells Estuarine Research Reserve. And we placed clams at 2 different tidal heights, an upper intertidal and a lower intertidal, in May of 2014 and again in 2015. And we had several, 5 different treatments, from open controls to something called flexible netting. I'll show you pictures of these in just a second. Netting with a piece of Vexar, which is a heavy duty piece of plastic that has an aperture of about 1/4 of an inch to try to keep crabs out. And then I discovered something called pet screen. And pet screen is made by a company called Pfeiffer, and it has a 1.7 millimeter by 0.9 millimeter mesh. So it's pretty small. And I didn't think that it was going to work, because I figured that it would get clogged, or the flow rates would be so low that nothing would survive. But I was surprised. These are what the controls look like. So these are exposed. These allow predators to prey freely on the clams inside. This is an exclusion net that's a 1/6 of an inch. This is the same exclusion net, but I've wrapped it around a piece of Vexar to try to keep it in place so that I'll keep out crabs. And then this is the pet screen. So the pet screen was used on the top of some of the pots, and it was used both on the top and an insert in the bottom to try to keep out milky ribbon worms. So when we went back in October to look at how well everything did in each of the treatments, we found many of our hatchery clams survived, but that's not really what the story is here. We found a lot more of other clams, wild recruits, more so than we did the hatchery clams. So notice that none of these have that hatchery mark, and these are wild clams that we found in those pots. So some of the pots, when we took the cover off, they looked like this. They were just brimming with extra clams. And here's an example of one of the replicates that we had put out, this one in the Webhanet, where we got somewhere around 250 clams extra from the treatments. So when we look at what we found in the Fore River during that time, the first thing to notice is that we found few. These are young of the year or small clam recruits. We found very few at the upper. It didn't make any difference what the treatments were. In the lower, we found some, but primarily they were in the pet screen, the treatments. And we found almost exactly the same thing at the Webhanet. So when an ecologist sees a pattern, they usually take advantage of it and try to ask more questions. And one of the questions that I asked from this study was, could I replicate what I saw in another place using a different philosophy? And what I did was come up with this wooden box. It's just a 1 foot by 2 foot by 3 inch deep box made out of spruce strapping. And I stapled pet screen on the top and on the bottom, and then just put lath over the staples so that they wouldn't dissolve or anything. And the boxes are empty. And then I just placed the boxes on top of the mud flats and left them there, anchored at each end with a lath that I pushed down into the mud, and then used a couple of galvanized nails to pound the edge of the lath into the box. So these are passive collectors. So clams are settling on the mud flats, and then they'll settle into these boxes and hopefully be protected. So this is kind of a predator protection area, but for smaller clams. We placed groups of 6 of these in 20 different places in the Harriseket River in April of 2015. So 120 boxes. And in some of those boxes, we found as many as 1,600 clams, or over 1,000 clams per square foot. This is a very happy clammer with some of those results. And this is just a quick schematic of what we got for results. That side of the river that's labeled wolf or wolf neck side is the eastern side, and that's the area where most of the clams that we found were occurring. And it's also the side of the river that's been not shut down by anything other than the lack of clams. And so this is where we found most of those softshell clams. We've done various studies using these recruitment boxes, and one of the studies that we did looked at different kinds of controls. So one could argue that we're not really seeing an effect due to predation. We're seeing an effect because the box somehow is attracting these clams. So this particular, and I'm almost done here, this particular slide shows what we find when we have controls that have open tops and different kinds of bottoms, and when we have mesh tops that some of the mesh is actually missing, 1/4 of the top is present, or 3/4 of the top is present. And so this is a pretty good example of how the boxes really are working. They're actually passive collectors. And this led us to be able to set up a softshell clam recruitment monitoring network funded by Sea Grant. And 2020 was the first year. We've divided the coast into 3 regions, looking at Wells, Scarborough, and Brunswick in the south, Wiscasset, Bremen, and Islesboro in mid-coast, and Frenchman's Bay, Beal's Island, and Sabayak down near Pleasant Point in the down east area. And the recruitment network is designed to increase visibility about this fishery during this time of changing marine environments, and to create a data set so that shellfish managers and others can understand the factors that affect the fishery. And then finally, we encourage participation by clammers and shellfish committee people and other municipal officers, and certainly K-12 students and teachers. In 2020, we set this out with the good intention of having everybody help, but of course, because of COVID, we didn't get a lot of participation. And we're hoping that 2021 is a better year for that, at least hopefully in the fall. But we're going to move on and use this network to go forward to see if we can make some predictions about what's coming in the fishery. So I guess that's it. I almost finished in time. Sorry, I went a few minutes over. And I guess it's time to answer some questions.
That was perfect.
So how do we do that?
That was perfect timing.
That was awesome. I think my sound might be a little weird tonight. So Jake, maybe I'll let you spearhead some of the questions.
Yeah, I'd be happy to. Does anybody have their hands raised, or not at the moment?
Let me check.
Okay.
I don't think any hands raised yet, but we definitely have questions in the chat box.
We do, and I'm happy to go through them. So Brian, one of the questions from Doug was, do clams have to be in mud to develop into adults? Could they potentially be suspended?
That's a great question. It turns out that clams can be out of mud until they're about 1 inch in size. And then after that, they don't get much larger in length. but they kind of grow in on themselves. They look like miniature footballs after a while. So they can't without some pressure. Now, is it possible to grow them with pressure in the water column? And the answer is yes, you can do that. We just need to figure out, I mean, we've done it on a small scale, and we've actually kept clams alive for over a year in what we call clam sandwiches. We've just taken a piece of mesh and then a piece we sandwich clams between mesh, and then between vinyl coated trap wire and vinyl coated trap wire. And then we use nylon cable ties to keep the wire cinched up so that there's pressure on the valve. Without that pressure, they'll just die.
Great, that was a good question. We have a question from David. What mesh size protects the clams from predators? Or I'm sorry, protects the clams from crabs specifically?
Okay, any size mesh, probably less than a quarter of an inch to and less is probably the most advisable. Back in the 90s, we did some studies looking at how mesh size affects survival and didn't find much of a difference. But now that there's more green crabs around, and green crabs, when they settle, can actually settle into those recruitment boxes. Remember, that was 1.7 by 0.9 millimeters. So when green crabs are settling, they're very, very tiny and can fit through a 1 millimeter mesh. So the smaller the mesh, the better the protection. You're not going to be able to use the PET screening on a large scale. It's fairly expensive, but you can buy rolls of the 6-inch mesh pretty well. And if you need an address, just email me at bbeal@maine.edu, and I'll get you that information.
Thanks, Brian. Maybe Lander can include your email for contact in the
Sure.
email. Great. So we have another question from Doug. Is there data on clam shell thickness versus survival? Is the increasing acidity causing any current decrease in survival?
Okay, that's another question. When I got a chance to visit with the clammers down in Freeport in 2013 and 14, they were worried and concerned about ocean acidification. And that was the major hypothesis of why there weren't any clams, especially in that area on the eastern side of the Harrisicot River. So we set out to try to find out what the major cause was, and we found that other folks, not only in Maine, but in Washington State and in Oregon, had been using various ground up shells placed on flats to try to buffer the sediments. Certainly the sediments are acidic, and that's no question about it. But it turns out that the flats have been acidic for a very, very long time, and the clams have actually evolved a mechanism to try to produce shell in that fairly low pH environment. Here's how we know of that. We set out a study where we took ground up clam shells and we placed them into plots that were 10 feet by 10 feet. And then we put either no shell in some plots, 13 pounds of crushed shell in others, and 26 pounds of crushed shell in others. And so there's 3 treatments. And then we had another 3 treatments where we put netting over the 0 or over the shell or over at 13 pounds and over the shell at 26. And we let them, we did this for 3 years in a row. And what we found is that if the plot didn't have a net, we didn't find any clam recruits. So what it showed us then is that predation, at least now, is much more important than coastal or ocean acidification. I'm not denying or trying to reduce people's ideas about the effects of ocean acidification. We need to be concerned about it. But right now, the major threat to softshell clams is not ocean acidification, it's predation and green crabs and seawater temperature warming.
Thanks for answering that, Brian. We had a question from Sarah. Did you see any difference in the recruitment boxes between ones that were lined with landscape fabric and the ones that were not?
Well, I didn't even talk about landscape fabric, so I know this must be Sarah Randall from Freeport, my co-PI on the Sea Grant project. So what we found is that sometimes there are environments where when we place these boxes on the mudflats and come back 6 months later, there's a gap, sometimes 1 inch to 3 inches between the bottom of the box and the mudflat surface, where the tides and other things have kind of carved out or eroded the mud underneath. And so it turns out that you can fit about 50 clams when they're settling into one of those meshes, one of the meshes of the pet screen. So if a clam goes through the pet screen and then goes to the bottom of the box and there's nothing there like mud sticking up through the bottom because it's eroded, then we may lose those clams. So what we've done in some of those erosional environments is to place this ground cloth. It's like a weed preventer that people use in their gardens. It's this woven polypropylene fabric. It allows the water to seep through very slowly, but it retains the clams in the mud. And so in places where there's been erosion, we find tremendous differences in the number of clams between the pet screen boxes and the fabric bottom boxes. In fact, there are some places where we won't put the pet screen bottom and top boxes out anymore. We'll put the pet screen top with a fabric bottom out exclusively.
It's a very good example of some awesome problem solving right there, and trial and error. We have a question from John. This is an interesting question. If in a condensed area, when one clam spawns, does it trigger a mass spawn?
Ah, boy, these are fantastic questions.
Got some good viewers here.
Well, you know, it's hard to see what's going on in nature. And so when we bring things back in the lab, we make some, you know, adjustments and we make some predictions about what might be happening. So when we bring clams into the hatchery to spawn, typically nothing happens until a male clam spawns first. And that male clam, and all clams have an internal hormone called a pheromone. And that pheromone is emitted when the sperm is released from the males. And it's kind of a signal that says, yes, it's time for everybody to spawn, because, you know, evolutionarily speaking, it doesn't make any sense for an individual to spawn and that's it. It's a waste of energy. So great question. We think that spawning occurs through this pheromone transfer to other clams. Yes.
Thanks, Brian. We have another question from John, actually. Oh, I'm sorry, different John. If higher water temperature leads to more green crabs and more depredation, is the loss of the population inevitable, or is there some sort of adaptation that might occur?
Well, the problem is that green crabs are an invasive species, and so adaptation is really occurring on an evolutionary scale, and softshell clams don't have time for that. Green crabs have been in Maine since 1905. They've been in North America since 1817, and while there has been some adaptation, I mean, clamshells in 2019 are probably similar in many areas to clamshells in the early 1900s. Clamshells actually do vary. You have thicker shells in more gravelly environments than you do in muddy and sandy environments. So even, you know, clams are fairly well adapted, and that allows them to be fairly ubiquitous in the soft bottom intertidal. Is the demise of clams inevitable because of the increasing water temperature? Well, the answer is yes. I think it is, but I do think that we have some things that we can try to overcome or swamp out the effects of predators. And this gets to a testable hypothesis that the recruitment boxes will allow us to do. So here's the deal. You know that lobsters have a minimum size and a maximum size, but do you know why they have a maximum size? Well, they have a maximum size because the relationship between the size of the lobster and the number of eggs that females produce is exponential. So animals So we don't fish lobsters greater than 5 inches carapace length. And so when you look at a lobster that might be 25 pounds, it has a tremendous number of eggs, much more certainly than a lobster that's 1 pound. But when you look at the relationship between size and eggs, it's not just straight, it's actually exponential. So it turns out that many bivalves, oysters and mussels and softshell clams, have the same sort of relationship. So clams do not do what you and I do, which is undergo reproductive senility as we get older or larger. So clams continue to breed into old age and produce exponentially more eggs or sperm. So there's an idea that if we set an upper size limit on softshell clams, that there might be more eggs and sperm and ultimately larvae that can settle to the flats produced. Now, it's a hypothesis. Another way of increasing the number of clam eggs or larvae in the water column is to say, all right, well, if you live from Freeport to Phippsburg, and we know that the water temperatures are going to be 50 degrees for the next week, that's a temperature that triggers spawning. So let's shut down the clam fishery for that period between late April and early, a week into May, and allow whatever clams are in the mudflats to spawn. So, there's some problems with that in terms of the fishery and the people that catch clams and dig clams for a living. But John is right. If we don't do something and seawater temperatures continue to increase, green crab populations are just going to keep on getting greater and greater unless something else comes in that eats green crabs. I don't know what that is at the moment. But my point with the recruitment boxes is we're going to have several years of, if you will, baseline data from at least 9 communities and 2 mudflats in each of those communities. So we've got 18 study sites, and we're always looking for more. And if something were, if a rule was adjusted so as to put in an upper size limit or to create a spawning, delay, if you will, don't fish while they're spawning, we could actually use these recruitment boxes to estimate the impact on settlement. So anyway, that's something.
We're going to have to make green crab soup more popular.
Well, and people are doing that. But the problem is that you're not going to be able to fish down the green crabs. And the reason is because they have such high fecundity. They produce, 1 2-inch animal produces 165,000 eggs. And the traps that are used are going to allow small crabs and small lobsters, they have to because of the lobster rules, they're going to allow small lobsters to escape. So the only crabs that are being caught are, the adults, which is fine, but where the rubber meets the road with green crabs and softshell clams is not a 2-inch green crab. It's a 5 millimeter or 3 millimeter or 2 millimeter green crab. Those are the ones that are having the enormous effect on the population. And a fishery isn't going to take any of those, might have a marginal effect, but not real.
Thanks, Brian. I just want to check in with Lander and see if anybody has their hand raised.
I'm still not seeing any hands raised.
That's all right.
Have we lost most of the people? I can't tell.
No, actually, we still have the majority of our viewers still. And we have 4 minutes. So if you want to raise your hand and ask your question directly to Brian with your audio, you're more than welcome to. Brian, we have a couple more questions for you in the chat box to keep us. Let me see. Let me just stay organized here. Here's an interesting question. Do you see any effect on the increased southwest temperatures on clam shell structure?
That's very similar to that question about thickness and ocean acidification. And I'm not quite sure I understand the southwest what temperature?
Yeah, maybe I'm making assumptions, but it just says SW in caps. So yeah, maybe I'm
water.
Oh, seawater?
Seawater?
Oh,
seawater temperatures increase?
Maybe. Yeah, so I guess that's a similar question.
Yeah.
Who is that from? David, if you want to offer any clarification in the chat box, please go ahead. We've got some time to ask your question for you. And I think there was one more. Let me just see. And then there was another inquiry about the water acidity in terms of the temperature. And I think what Doug is asking is one incredibly more, detrimental to the species than the other. And I think you kind of already touched base on that, but.
Yeah, I mean, certainly ocean acidification is having an effect. Is it the reason why the clams have declined? And I think that we've answered that in many different areas when we've either planted small clams or we've looked at recruitment and found that, if you don't protect, you don't get clams. And one of the things that I failed to mention about that buffering study was when we put the nets over the shell or the nets over the non-shell treatment and took pH measurements, we actually found lower or more acidic sediments under the nets than we did outside the nets.
Wow.
Yeah, that was an eye-opener.
Yeah, absolutely. We had a question from Doug about, do you think that commercial scalloping on lines with suspense scallops from tiny holes in the shell near the joint can be successful in Maine?
Well, that's a great question. I'm also interested in scallops. I think that scallop industry for aquaculture is going to need more than a collection of wild animals, which is how everyone that's raising scallops right now is getting their seed. There are some places that consistently get, hundreds to thousands of scallop seed, but it's not an industry because it's mostly a small scale, a few ropes and things like that. If we're going to talk about a scallop farm or a scallop industry, we need to be able to do something that we've not been able to do in 30 years at DEI and in other places, and that is to develop a hatchery stock of seed. For whatever reason, scallops are very difficult to raise. They have a 35 to 40 day larval cycle that we can't reduce in the hatchery because if we increase the water temperature too much, they will die. And we've failed time after time in trying to get these things to a seedable size. So yes, I think that putting scallops and drilling holes through their shell or their hinge and adding them to ropes, they grow and they survive. It's just the numbers, and the numbers are very, very small at this point in time. I'm hoping that a scallop aquaculture industry can develop in time.
Thanks, Brian. Jake, we do have a hand up now.
Oh, that's awesome. Great. Go for it.
David Porter has a question. So David, I'm going to allow you to speak, and you just have to unmute yourself.
Hi, can you hear me?
I can, David.
Yeah, I have a question about the size of the predator crabs that you talked about being very small, and yet the large scale nets that you put over clam flats increased clam productivity. Mm-hmm. But so that's not the smallest crabs that get into it's larger crabs that are being excluded.
So certainly larger crabs are being excluded. So when we found that 1,300 or almost 1,400 clams per square foot, wild clams under those nets, we did find a few small crabs, but certainly not enough to make a dent in that particular density. So those nets, you're absolutely right. They're not going to exclude every crab. It's not perfect.
So on a commercial scale, you can we could put out nets like that in a flat where there haven't been any clams, and it's different from your little boxes that you put
Yes.
out with a finer mesh net. Yeah.
Mhm.
Okay.
So, yeah, and we've done, so one of the things that we've asked, working with communities is, can you just simply put out a net
Right.
and hope that you get, clams to settle? And we did it with the biologists with the DMR. We worked in Jonesboro, we worked in Booth Bay, and one other site, gosh, I'm having a hard time remembering. Booth Bay, Jonesboro, and one area in between, the Georges River. And what we found was that less than 10% of the nets actually produced clams the way we had hoped. It was very disappointing. So it's kind of hit or miss is basically what we're saying.
Do you put the nets down with staples, like garden staples?
No, usually we're working in an environment that's soft enough so that we can walk on the edge of the net, and our bulk, some of us are bulkier than others, forces that net down into the mud 6 to 8 inches, and then we kind of backfill with our foot. In other places where the sediments are a little more, they're more coarse, we actually take clam hose and dig a furrow around the entire perimeter, down about 8 inches, and then tuck the net in and then backfill again by moving that mud that we've just dug in Gomer pile fashion back into the furrow that we've dug. One of the things that we've done in 2017 was to take those 1 foot by 2 foot boxes and expand them to 4 foot by 8 feet and place them on top of a mud flat. And what we found, and those boxes are 16 times larger than those smaller 1 foot by 2 foot, and we found 16 times more clams in those large scale boxes. So that might be something to try out. And, putting out a couple of recruitment boxes in an area gives you an idea of, is this an area that we're getting recruits from? Because, just because you put a recruitment box out doesn't mean that you're going to get, softshell clams in them. This past year, along that network, we didn't find anything dramatic in 8 of the 9 communities. Dramatic being, clams in the thousands like we'd seen in the past. We were sometimes getting 150 to 200 clams. Some of the ones had 0. Some of them had 20 or 30. There was just one site in far eastern Maine where we averaged almost 1,300 clams per box. So the boxes can be used as seed, as a valid seed source. But I do it on a small scale first and put boxes in different places. And that's basically what the recruitment monitoring network is all about, is finding these areas and seeing if there's any consistency from one year to the next.
Those recruitment boxes aren't for growing commercially sized clams. They're just to see if there are clams that settle there.
Exactly.
Yeah. Thanks.
You can't grow clams. You need at least 5-inch deep vessels to grow clams to at least 2 inches.
Thanks, Brian.
Sure.
Yeah, thank you, Brian. I have 6 after, so I just want to check in with Lander and see if there's anybody else that has their hand raised. We might give them a quick opportunity to ask their question.
Don't see any other hands raised. It looked like there were 2 more questions in the chat box, so I don't know. I know, Brian, you've been talking a lot for the last hour, so if you want to take those 2 last questions and then we can wrap up.
Absolutely. I talk for a living. I teach, so I don't get tired of it, especially when it's about clams.
Well, you're very good at it. This is really enjoyable to listen to.
Thank you so much, Brian. So we had I think I missed a question. It was a pretty general question up a ways up in the chat box, but how large could a clam grow? I guess an open-ended, like, general statement.
Yeah. So I have one of the biggest clams that I've ever seen as a shell in my office, and it's about 6.5 inches in size. I've rarely seen clams larger than that. I've heard from some clammers that they've seen clams that might be 7 to 7.5 inches, but that's a pretty big and old clam. Yeah.
Cool. Okay, so maybe there are 3 questions actually, because Jake caught that one further up. So now 2 questions. There's another one from John. Is there any DMR or legislative momentum for the size limit?
None.
Cool, that's an easy one.
Yep.
And then John had another question. How does a green crab attack a clam, and is the success dependent on clam size?
Oh, very good. Yes, the success is related to clam size. So clams actually burrow for a reason, and they burrow to escape their predators. And so there's a depth refuge from most predators for clams. And how are clams found by crabs? It's by scent typically, and the green crab typically will take a hold of well, actually, we had some pictures. Let me, well, never mind. But one of the shots that I had was showing actually a shell in fragments, and how that crab had glommed onto that was by crushing the outer margin. It's where the newest shell is, and it's the easiest part to actually pry. Once that crab has crushed part of the shell so that it can essentially get into the inside of the clam, it then can just use its claws to keep on clipping and teasing out the mesh until it might get to the adductor muscle, the muscle that actually keeps the 2 valves tight together. And so it gradually is able to get and cut that adductor muscle. And at that point, there's actually 2 muscles, but all it needs to do is to cut one, and that frees up that clam so that it's wide open. So it starts on the margin of the shell, works its way into the meat, and then to the adductor muscle and feeds on it that way.
Wow. That's amazing. Well, I think we might have gotten through all the questions,
Wow.
all the
I
amazing
think so.
facts.
And Brian, thank you so much for letting us share your email with folks with the follow-up email. So if anybody didn't get their question answered in the full capacity that they were hoping for, maybe you could reach out to Brian directly and ask.
Absolutely.
Thank you so much.
Okay.
Thank you so much for being with us, Brian.
Sure, and thanks for the invitation. I appreciate it. It's been great.
It was awesome.
Absolutely. Have a great night, everyone, and look out for a follow-up email.
Thanks for tuning
in. Bye-bye. Thanks, guys. Thanks, Lander.
Thank you so much, Brian. It was great.
See you later.
See ya.
Bye-bye.